So in today's class, we discuss about the questions related to PYQs of 2023 paper. Okay. Here we see the question: number of base pairs present in DNA wrapped around a core of histone protein. So it is related to the DNA structure. You know, the formation of the nucleosomes, how they are formed. Once we'll see here, the DNA, double-stranded DNA, it is wrapped, particularly in case of the eukaryotes. This is related to histones. Histone proteins are present only in the eukaryotes and they are not present in the prokaryotes. So that is the main difference. Here in case of the eukaryotic DNA, the double-strand DNA, it wraps around the histone proteins. Coiling, it is also called as DNA coiling. So except in case of the mitosis or cell division phase, DNA is wrapped, it is coiled. So it coils around the histone proteins. You see H2A, H2B, H3, H4. All these are the histone proteins. There are 8 histone proteins in each nucleosome. So nucleosome is the structure. It is formed by the coiling or wrapping of the double-stranded DNA around the 8 histone molecules. That structure is, each structure is called as nucleosome. So all these nucleosomes, they form a supercoiled structure of a DNA. So the question is, what is the length of the DNA? So DNA length is measured with respect to base pairs. How many base pairs are there? It indicates the length of the DNA. So in each coil, so you see the 146 base pairs of DNA in the nucleosome. So that is the right answer here to the question. Number of base pairs present in DNA wrapped around a core of histone protein. So that is the question. Because the length of the DNA is measured in base pairs. So it is 146. So you should remember, in the nucleosome, the structure that is formed is called as nucleosome. There, 8 histone proteins are wrapped. The DNA between the two successive nucleosomes is called as linker DNA. Okay, so 146 base pairs length, that you should remember. And next, if a red flowered plants of Antirrhinum species are crossed with white flowered plants, then expected F1 progeny in incomplete dominance is. So this question is related to incomplete dominance. Okay, here there are two parents, capital, that is the homozygous dominant, and next parent is smaller, that is recessive homozygous parent. So the capital, that genotype, it produces capital as a gamete. Okay, and from smaller parent, the gamete is smaller only, one gamete with the smaller. Then in F1 progeny will be capital R and small r. But here, if the capital R is dominant, then the phenotype should be red color. Okay, but here it doesn't show the red color. Instead, it shows the medium color, pink color or purple color. So that means there is incomplete dominance. So you see the answer. Then what is the phenotype of the F1 progeny in incomplete dominance? Incomplete dominance, that means all pink flowers, all the flowers, they are not either red or white. It shows the medium color. That is the, because here in the incomplete dominance, no allele is dominant. Both are equally there. So that is called as incomplete dominance. The phenotype will be pink, all pink colors. You see once here, there is two terms in the genetics: incomplete dominance versus codominant. Other term also you listen sometimes, that is codominance. What is the difference between incomplete dominance and codominant? If there is total dominance, then the phenotype will be red color. In the incomplete dominance, you see the pink color. Instead of red or white, we see the pink color. And what is the difference between the incomplete dominance and codominance? So here, same, capital, smaller. One is red color, another is white color. The progeny, the F1 progeny will be like this. It is not pink, but some petals are red color, some petals are white color. That is the coda. That means both the genes are expressed, both the phenotypes are equally expressed. Okay, it is not a mix like in case of the incomplete. In incomplete dominance, it is a mix. It is not red, it is not white. It is a mixed color, pink. But in case of the codominance, because sometimes they may ask about codominance also. Here we see the both the phenotypes, red and white. That is the difference between the incomplete dominance and codominance. And now we'll see another question. A typical prokaryotic cell is found in. So prokaryotic cell, we don't find a distinct nucleus in the prokaryotes because there is no nuclear membrane surrounding the nuclear material. Okay, so nostoc, what is that? It is a blue-green algae. It is also called as cyanobacteria. It is the only algae with the prokaryotic cell structure. That is the main feature of the blue-green algae. It is also called as cyanobacteria because it resembles bacteria with respect to its prokaryotic nature. In case of the other algae, chlorophyceae, Pheophyceae, Xanthophyceae, Rhodophyceae, all these are eukaryotic except the cyanobacteria or blue-green algae. So nostoc belongs to cyanobacteria blue-green algae. So it is prokaryote. And others, Eleutheryx, Aspergillus, Vaucheria. Say Aspergillus, it's a fungi. Fungi also eukaryotic. Eleutheryx, Vaucheria, they are algae. They are also eukaryotic. So only prokaryotic one is nostoc. So that is the right answer. Next, a gene showing codominance has. So just now we discussed about the difference between the dominance. Okay, incomplete dominance and codominance. So what is codominance here? Alleles that are recessive to each other. That is not right. Both the alleles are independently expressed in the heterozygote. That is the F1 phenotype. In the F1 phenotype, that is a first progeny, F1 phenotype, we see in case of the incomplete dominance, we see the pink colored flowers. But in case of the codominance, we see both the phenotypes independently because both the genes, red colored and white color, both the genes are expressed. So we see just now we saw the here codominance, we saw the red colored petals and white colored. Both are independently expressed. So that is called as codominance. So that is the answer here. Both the alleles are independently expressed in the heterozygote. That is the answer. And next one, stipe, hold fast and frond are the terms associated with. So where we see this terms, stipe, hold fast, frond. In the mushroom also we see the structure stipe, but hold fast is absent, frond is absent. So where we see that, we see in case of the laminaria. Actually laminaria, it belongs to Pheophyceae. It is not given in our syllabus, but it was asked in 2023 question paper. Laminaria. sargassum, all these belong to Phaeophyceae. These are all sea algae. Mostly Phaeophyceae consist of sea algae. Okay, and laminaria, you see the laminaria in the structure. What is type here? Just once you see here. So hold fast. It is used for the attachment of the sub base. Okay, and stipe. This is the stipe, and this is the frond. In the structure of the laminaria, we find these terms. Okay, so that is the right answer is laminaria. Next, gametophyte of bryophyte is. So in case of the in algae, from the bryophyte onwards, we see alteration of generations. In the life cycle, we see alternation of generations. What does it mean? We see both gametophyte and sporophyte in the life cycle. Gametophyte is haploid structure, whereas sporophyte is diploid structure. Okay, and here in the bryophyta, gametophytes are dominant and sporophytes are not dominant. They are dependent on the gametophyte. Okay, they depend on they both, you see in the life cycle of bryophytes. For example, Markamtia. You see, Markamtia, it is a gametophyte. On the gametophyte, we see the formation of the archegoniophore, archisporiophore, and they form the after that they form the sporophyte on top of the gametophyte. And these sporophytes are diploid. They are produced after the sexual reproduction. And the sporophyte dependent, they are attached to the dominant gametophyte, and they rely, the sporophytes, they rely on the gametophyte for their food. So in bryophyte, what they are asking, gametophyte of bryophyte is green and vascular, that is one option. Independent multicellular, that is one option. Dependent on sporophyte, it is wrong. Okay, because gametophyte is dominant. Sporophyte is dependent on gametophyte in case of the bryophytes. Okay, but in case of the gymnosperms and angiosperms, okay, gametophyte is dependent. It is reverse. In reverse, gametophyte is dependent on the sporophyte. But here in the bryophytes, gametophyte is dominant. And next, gametophyte is dependent and unicellular. It is also wrong. It is not unicellular. It is multicellular and it is independent. Okay, and the right option should be, it is dominant and multicellular. But they gave independent and multicellular, so we can choose the nearest answer, that is independent and multicellular. Next, reticulate chloroplast found in. So we see the chloroplast in algae and in bryophytes, pteridophytes, and in the angiosperms. But since they are asking about the reticulate chloroplast, in our syllabus we have Ulothrix, Eidogonium, Spirogyra, Wachiria. You know, Spirogyra, the name is given due to the presence of the spirally coiled chloroplast. You know about the Spirogyra. But in Eidogonium, it is reticulate. Chloroplast is reticulate. You see the structure of the Eidogonium here. You see it's a single filamentous structure. It is in the form of Eidogonium. It belongs to Chlorophyceae. It is a filamentous structure, unbranched structure. In the cell, we see the nucleus, a single nucleus, and there are so many. Here you see the chloroplast in the reticular form. Okay, in the reticular form, there are pyrenoids also present in the reticulate chloroplast. Pyrenoids, they store the food in the form of the starch. Okay, so this is all about Eidogonium. So the right answer is reticulate chloroplast is found in Eidogonium. That is the right answer. Next, in one strand of DNA has nitrogenous base sequence as ATCTG. What would be the complementary RNA strand sequence? So if the DNA strand acts as the template for the synthesis of the RNA, for example, for the mRNA synthesis. Then, based on this sequence, DNA sequence, what would be the RNA sequence, mRNA sequence? That is the question here. So, since here there is A, the complementary base is T, but T is thymine is present in the DNA, not in the RNA. So, instead of in instead of AT, we have to say instead of thymine, in the RNA there is uracil. So AU, the first nitrogen base in the RNA is U, which is complementary to A, and there is a T in DNA. So here also, what is the complementary nitrogen base to T? That is A, adenine. So adenine is right answer. And C, what is the complementary one? G, GC. There are 3 hydrogen bonds between always A pairs with T, G pairs with C, and vice versa. C pairs with G or T pairs with A in reverse. So C means there should be G, and T, that means here is A, G, that means here is C. So that is the complementary DNA sequence. So this is the right answer. Since they asked about the mRNA, we should be careful. Okay, we should replace thymine with uracil in the RNA structure. Next one, which of the following is known as sac fungi? So in the fungus, we already discussed Phycomycetes, Zygomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. So all these are the different kinds of the fungal groups where we find sac fungi. That means in Ascomycetes, there is a sac-like structure that is called as ascus. Due to the presence of the ascus in the Ascomycetes, that Ascomycetes fungi is also called as sac fungi. So that is the right answer, Ascomycetes. Next, ATP energy is produced in the following cell organelles. Okay, so we know that ATP, the formation of the ATP is called as phosphorylation. We already discussed about it. There are 2 kinds of the phosphorylation. One is photophosphorylation, which occurs in the light phase of the chloroplast. Okay, in the light phase in the chloroplast. There are 2 phases in photosynthesis. First phase is light phase, which occurs in the chloroplast thylakoid membranes. That is called as photophosphorylation. During the electron transport, ATP is synthesized. That is called as photophosphor. But in case of the mitochondria, also there is a ATP synthesis. Particularly, the food material is oxidized. There are glycolysis, Krebs cycle, and electron transport chain. So in the during the electron transport chain formation, there is a process called as oxidative phosphorylation. That means ATP is synthesized during the electron transport, and the energy here is called as produced due to the oxidation of NADPH or FAD. Okay, FADH2. So due to the oxidation of these molecules, the energy released is used to synthesize the ATP. So it is called as oxidative phosphorylation, which occurs in mitochondria. So they asked about ATP energy is produced in the following cell organelles. Only chloroplast, it is wrong. Photophosphorylation occurs, but only mitochondria, it is also wrong here. Oxidative phosphorylation occurs. The right answer is chloroplast and mitochondria. In the chloroplast, there is photophosphorylation. In the mitochondria, there is oxidative phosphorylation. That is the right answer. During this phosphorylation, ATP is synthesized. And next one, an artificial group within fungi is. We already discussed. There is Phycomycetes, Zygomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. So deuteromycin is treated as an artificial group because there is no sexual reproduction identified in these organisms. So all the fungal organisms where sexual reproduction is not observed or not found until now, they are all grouped in the class that is Deuteromycetes. So it is also, so this is the artificial group. An artificial group within fungi is Deuteromycetes. That you should remember. And the correct statement for Spirogyra is that. So Spirogyra, related to Spirogyra, Actually, we have idogonium, but in intermediate you might have studied Spirogyra. So what they ask, filaments having scalariform conjugation are always homothallic. Filaments showing lateral conjugation are always homothallic. Filaments showing lateral conjugation are always heterothallic. And asexual reproduction is brought about by juice. So what they ask, the correct statements for Spirogyra, filaments having scalariform conjugation are always homothallic. What is homothallic? Homothallic means here the male and female gametes, if they are both produced from the same filament, from the different cells of the same filament, that is called as homothallic. You see here in the diagram, yeah, you see this is Spirogyra. Here we see the spirally coiled chloroplast, and here there are two types. One is homothallic, another is heterothallic. What is homothallic? Here there is two types of conjugations, lateral conjugation and scalariform conjugation. In the lateral conjugation, both the male gamete and female gametes are produced from the cells of the same filament, which are next to each other. One acts as the male gametangium, another acts as the female gametangium. From the male gametangium, male gamete is produced. From the female gametangium, female gamete is. So there is a lateral conjugation. Here, male gametes enters into the next cell, that is female gametangium, and it fertilizes the female gamete there. It forms the zygospores in the female gametangium. This is called as lateral conjugation. It occurs in the same filament, so it is called as homothallic. But in case of the scalariform conjugation, two different filaments, they come closely and they form the conjugation tubes. Here one in the one filament forms the male gametes and another filament forms the female gametes. So it is not called as homothallic, it is heterothallic. So in the scalariform conjugation, we see the heterothallic condition. That means the male and female gametes are formed in different filaments. They form the structure like this, so it is called as scalariform conjugation. So here the question is, filaments having scalariform conjugation are always homothallic. That is wrong. Filaments showing lateral conjugation are always homothallic. That is right. That means the lateral conjugation occurs in the same filament because both male and female gametes are produced in the same filament. That is lateral conjugation. Okay, so that is the right answer. Next, the rate of decomposition is faster in the ecosystem due to the following factors, except you should remember, understand that except means not that one. That means the rate of decomposition is faster in the ecosystem due to the following factors, except. That means where detritus rich in sugars, the decomposition occurs very fast because sugars are there, no microorganisms, they like sugars very much, so decomposition occurs very fast. But they are not asked, they are asking about the except. That means not the case. That means where the decomposition is slower. That is the question. They are asking except means where the decomposition is slower. If it is detritus means the decomposing organic matter. If it is rich in sugars, the decomposition is very fast. And next, warm and moist environment. Yeah, if the environment is moist and warm also, the decomposition is very fast. And next, presence of aerobic soil microbes. If the aerobic soil microbes present also, decomposition is very fast. So finally, you see the detritus, that means decomposing organic matter, if it is rich in lignin and chitin, the decomposition is very slow because it is very difficult. Lignin is a very complex material. Chitin is also very complex material, which is present in the you see in the xylem. Okay, in the sclerenchyma, the walls are made up of lignin. So it is very difficult. It takes a lot of time for the decomposition of the organic matter with the lignin chitin. So that is the right option because they are asking about the slower decomposition. Next one is match list 1 and list 2. So this also they may ask. So you see in the left side list 1 oxidative decarboxylation, where we find oxidative decarboxylation between the glycolysis and Krebs cycle. The end products of the glycolysis are pyruvates. Okay, two pyruvates are produced after the glycolysis. From one glucose, after the glycolysis, two pyruvates or pyruvic acids are produced, and they undergo oxidative decarboxylation with the help of the enzyme called as pyruvate dehydrogenase. So A is connected to 2 here. And glycolysis, what is glycolysis? Here EMP pathway they are. Emden, Mayer, Huff, Parnas are the three scientists. They explained about the glycolysis. So it is also, glycolysis is also named as EMP pathway. So fourth one, for the glycolysis, EMP pathway is the match. And next, oxidative phosphorylation, where we find, we already discussed about the glycolysis Krebs cycle. Next, third one is electron transport chain, where oxidative phosphorylation takes place. So for this, the right answer is electron transport system, where we find the oxidative phosphorylation and synthesis of the ATP. And fourth one, tricarboxylic acid cycle. Krebs cycle is also called as tricarboxylic acid cycle or citric acid cycle. So here you see tricarboxylic acid cycle means the first one, citrate synthase. We find this enzyme in citric acid cycle or tricarboxylic acid cycle. So the here matching is A here, A with the two oxidative decarboxylation related to pyruvate dehydrogenase, and B4, B means glycolysis is EMP pathway, and C3 means oxidative phosphorylation is occurs in electron transport system, and D is 1, that is tricarboxylic acid cycle is in citrate synthase. Okay, it is involved in tricarboxylic acid, citric acid. So this is the right option, first one. Next, all given statements stand true with respect to lichens except. So that is the question. Okay, this is regarding lichens. So these are indicators of sulfur dioxide pollution as shown by their abundance in these areas. So they are asking all given statements are true except. That means we have to find the false statement. So lichens, they don't grow here. They are not the indicators of the sulfur dioxide pollution. Breathing pores help in gas exchange. These are found on the lower surface of the thallus. So in the lichens, in the lower surface, we find the breathing pores. They are useful for the exchange of the gases. So that is right one. So we should not choose that. We have to choose the wrong statement. This association is also termed as helotism. What is this association? The lichen association. In the lichen, what we see, the association between the algae and fungi. What is helotism? Helotism, in case of the symbiosis, what is symbiosis? Here in the lichens, algae and fungi, they have mutually beneficial relationship. That is called as symbiosis. But here, symbiosis also not truly symbiosis here. That is called as helotism. In symbiosis, there is symbiosis. It is mutually beneficial. But here in lichens, the fungal partner dominates the algal part. Okay, it forms a trap surrounding with the help of the mycelial filaments, fungal filaments. It forms a trap around the algae and it dominates the algae. So this kind of relationship is called as helotism. So this is not the true symbiosis. In the symbiosis also, the fungal partner shows the dominance. So it is called as helotism. that is true. So we should not choose this because we have to choose the wrong statement. This is the right statement. Next, fourth one, crustose lichens are pioneers of rock suction. We already discussed in the xeroxere or lithosphere. We see the rock succession. Rock is degraded due to the growth of the lichens. So crustose lichens, the succession starts with the crustose lichens. So they are called as pioneers of rock. So this is the right statement. So we have to choose the wrong statement. That is, lichens are indicators of the soil SO2 pollution. That is wrong state. They are not the indicators of the SO2 pollution. So next, in bryophytes and pteridophytes, transport of male gametes requires. This is very important question because usually bryophytes, particularly, they are also called as mosses. They grow very near to the water surroundings, in the marshy soils, in the wet soils, in the wet areas only these can grow. Why? Because for their fertilization, they release the anthrozoids. What are the anthrozoids? Anthrozoids are the male gametes with the flagella. For the movement of the flagella of the anthrozoids, they require water or moisture. Then only they can reach the archegonium. Okay, the male gametes or anthrozoids which are released from the antheridia, they reach the archegonium with the help of the moisture in the atmosphere. So what they require for the movement of, for the transport of the male gametes, what they require? They require water in the form of the moisture. So that is the right answer. Not insects, not birds, not wind. In case of the bryophytes and lower pteridophytes, they require water or moisture for the fertility, for the movement of transport of the male gametes or anthrozoids. Next one, movement and accumulation of ions across a membrane against their concentration gradient can be explained by. Okay, we already discussed during the transport across a membrane, living membrane, the ion transport or anything. So if the one side there are more ions, in other side there are less ions. Usually ions move from the high concentrated area to low concentrated area through the membrane. It does not require any energy. That is called as passive transport. The transport which occurs without use of any energy, because from high concentration to low concentrations ion move. It is a natural process, so it doesn't require any energy. You see, if we drop a ball, it automatically falls down due to the gravity. See, there is a water in the tank. Okay, that there is a water in the tank and it flows down through the pipes automatically. It does not require motor. But if you want to lift the water from the underground against the gravity, then we require the energy. Like that, here, if we want to send the ions against the concentration gradient, what does it mean? Instead of from higher concentration to lower concentration, if we want to send it opposite the concentration gradient, that means from the low to high, then we need to use the energy. That transport is called as active transport. That means it involves the utilization of the ATP. We need to provide the ATP for the active transport against the transport transport of the ions against the gradient. That you should remember. Okay, for the transport of the ions against the gradient, concentration gradient, we need to give the ATPs. They require ATPs. The transport is called active transport. Next, in gymnosperms, the ovule is naked because, see, in gymnosperms, we don't find the fruits. We only find the seeds. Why? Because if you understand, the ovary contains the ovules. After the fertilization, the entire ovary becomes the fruit, and the inside the ovules they become the seeds. But in case of the gymnosperms, there are ovules, but there is no ovary surrounding the ovules. Since there is no ovary wall, there is no fruit. So that is the right answer. Ovary wall is absent, or ovary is absent, or ovary wall is absent in case of the gymnosperms. There are only ovules. In case of the angiosperms, we don't find the ovules outside. They are within the ovary. But in case of the gymnosperms, ovules are not surrounded by the ovary wall. They are not within the ovary. So only we find the seeds without fruit formation. So that is the right answer. Ovary wall is absent in gymnosperms. Next, in protein synthesis, the initiation codon is. So we already discussed about the initiation codon, that is AUG. There are three termination codons: UAA, UGA, UAG. These three are called as termination codons or nonsense codons. The initiation codon is AU. So these are very important. What is the termination codons? Also, you should remember which of the following genus is associated with the coralloid roots. So they are asking about the coralloid roots, where these are present in cycas. So that is very important. We know that cycas has a coralloid roots. They have um, blue-green algae or cyanobacteria. Nostoc and Anabina is present. These two, blue-green algae or cyanobacteria present. They have the symbiotic relationship with the coralloid roots. That you should remember. Next, the principles of numerical taxonomy were developed by numerical. We know that in taxonomy there are different types of the aspects: embryological relation of the taxonomy, numerical taxonomy. So it is a quantitative relationship. Who developed? So based on the similarity, it is a similarity index among the different species. Based on that, they form different groups, different taxa, different species, genera, all orders, series. All these are formed based on the similarity among the species. That is a similarity index. One analysis called that cluster analysis. Okay, dendrograms. They form the dendrograms, cluster analysis, to find out the similarity index. What is the how much it is similar? It is a quantitative measure. So Sneath and Sokal, they both developed this numerical taxonomy. Sneath and Sokal, you should remember that name. And which of the following features would distinguish a monocot from a dicot plant? So to distinguish the monocot means it has single cotyledon. And in the monocots, parallel venation is present. These are the two important things.